All right, everyone, it's showtime. Hello. Welcome to Florida Atlantic University's Harbor Branch Oceanographic Institute and the John and Barbara Ferreira Ocean Science Lecture Series. Many of you know me by now. My name is Dr. Gabby McHenry, and I am the Director of Education and Outreach here, as well as your host. As many of you know, this program has been a core part of the Institute's outreach for nearly 30 years, and we share some of the amazing work that's underway here and around the world. So thanks so much for being here today. We hope you enjoy the lecture and, of course, learn something new. Today marks our final lecture of the season, and what a season it has been. We welcomed record-breaking attendance and hosted 11 incredible presentations that explored topics from sea turtles to harmful algal blooms, discovering medicines from the deep sea, and even training the next generation of scientists and stewards. So whether you've been to one lecture or many, we'd love to hear your feedback. We're going to be sending you an email survey, and we hope you'll share what you enjoyed, and even some suggestions of topics you'd like to see in the future. I take all of these things into account when I put the schedule together, and a couple of our lectures this season were actually based on requests that you all gave us last year. So thank you for your feedback. Keep an eye out for that in your inbox. I'd also like to acknowledge everyone on our team who has worked so hard to make this season a huge success, from the faculty and the students who presented to over a dozen different staff members from multiple departments here who all work together behind the scenes to put these lectures on every single week. We couldn't have done this without them, so I'd like to take a moment to thank them all for their help with the series. Thank you. As always, I'd like to extend a very special thank you to our benefactors, John and Barbara Ferreira, and to all those attendees who have made gifts to support our Ocean Science Lecture Series. Community support has been the backbone of this program for decades. Whether it's your participation each week, sharing what you learned with your family or friends, or helping us to support the series, we appreciate you. And we hope that if you enjoyed the 2026 season and spending time with us each week, or you were inspired by any of the research or education that you heard about, we hope you'll consider making a donation to one of our programs, and you can see some more information on the slide behind me and chat with us after the lecture if you'd like to learn more. And if you're looking for a very fun and meaningful way to stay connected with us after the series is over, I'm excited to announce our 4th annual Ocean Film Festival, which we'll be hosting here this June. It's become a favorite event within the community, and last year we sold out of tickets for the first time ever, and we raised over $7,000 to support students here. So it's a really fun event. We show a variety of films curated by filmmakers from all around the world, and we tie in with some of the research that we do here. So definitely mark your calendar, and we hope to make this year's event a huge success, and we'll be sending more information by email on that as well. Okay, back to today. Please also take a moment to ensure that your cell phones are silenced. Thank you. Last time. As always, our presentations are also available virtually, so hello and welcome to everyone who's tuning in on our live stream, and they're recorded, so if you missed any presentations throughout the season, or there was one that you really loved and want to watch it again, you can do so on our website. For everyone here in person, we'll be hosting a meet and greet reception with our speaker and some cookies after in the gallery. We'd love for you to stay and strike up a conversation with our scientists, staff, and students to learn a little bit more about what goes on here at FAU Harbor Branch. While you're there, and I saw many of you already did, be sure to visit Jen at our pop-up gift shop and grab some flyers about our various programs. We'll also have our Discovery pontoon docked outside, and Sam will be giving a demo of our onboard tech, which includes a hydrophone that Matt will talk more about today. So if you want to see that in action, pop outside and chat with her after you grab a cookie. And that brings us to today's lecture, which is titled Breaking Ground with Underwater Sound by Dr. Matt Ajemian, who is an associate research professor and has been at FAU Harbor Branch since 2016, where he leads the Fisheries Ecology and Conservation Program. Matt is a marine ecologist who studies how various sharks, rays, and bony fishes interact with their environment as well as fisheries activities. He has published 80 articles in the scientific literature and has been awarded competitive early career awards through both the National Academy of Sciences and the National Science Foundation, as well as applied grants from NOAA Fisheries, Department of Defense, and Florida Sea Grant. While Ajemian has worked with a wide range of species and scientific questions throughout his career, he's most well known for his work on ray-shellfish interactions and designing technologies to capture and understand these elusive interactions. And we're going to learn all about that today, so join me in welcoming Matt.
All right. Good afternoon, everybody. Thanks for sticking around through the last lecture, and really excited to present this really interesting work to you guys. It's been a dream to kind of answer some of these questions ever since I got my PhD, and I was very fortunate to learn some new tools, which I'm still learning, and to get back to some of these questions with kind of a new approach. So for those of you that know me and know my lab, you may think of us as fish people. Not literally like human fish, hybrids, but we study a lot of fish, and that includes sharks, rays, and bony fishes. We've worked on them offshore and inshore, and this is just sort of a smattering of the species, and a lot of our work is very applied and looks at fishery interactions and things of conservation consequence. So that's been a lot of our work, but you may not know that I'm really interested in predator-prey interactions as well. And I'm sort of a wannabe shellfish enthusiast. So I like eating shellfish. I also like just going out and shelling at the beaches here. And we're trying to get a little more involved in the science behind these animals too, particularly because a lot of what we study eats shellfish. And so shellfish in general, these are really important species. These are generally your mollusks, right, which include your bivalves, your gastropods, which are marine snails like conchs, even some squids and octopuses, but also crabs. And so these are responsible for providing a variety of ecosystem services to our various marine realms. And they're really crucial to the ecosystem health of these habitats. And so some things that shellfish do is transfer nutrients or facilitate transfer from land to other organisms. They can also store carbon and fix nitrogen. These are important things for nutrient cycling in our ecosystems. They provide tremendous habitat by also stabilizing shorelines and creating nooks and crannies, kind of like this oyster reef does here. And that also helps increase complexity of the habitat, makes little homes for other things. And again, my favorite part is shellfish are food for other types of organisms, so both marine and even terrestrial. They really facilitate the transfer of energy from things living in the seabed to animals that are what we call more pelagic or mobile and swimming above in the water column. And commercially, obviously, they're really important too. So how many people here enjoy eating shellfish? Okay, yeah. You can be selfish about shellfish, that's cool. All right, so human consumption, I mean, we have a global market value in shellfish. This is dated, so it's probably a lot more these days, of almost 24 billion. And so what do we eat? We eat bivalves, we eat gastropods like conchs and other snails, and hundreds of species of each of those. But you may not also realize that these animals also create important materials for construction, such as lime and shell hash. Anybody here have a shell hash driveway or walkway? Yeah, that all came from one of these organisms at one point. And some of that's even ground to grit for poultry supplements. So there's some chickens doing their thing, eating rocks. Okay, so, and then heritage-wise, these organisms obviously have important culinary and ecological importance to various cultures around the world. You can't really go a season without skipping by a seafood festival in a coastal town. So lots of importance there. And unfortunately, shellfish are on the decline in many different areas. They're threatened by numerous things, including overexploitation and overharvest, meaning we're taking too many without leaving enough there to replenish themselves. Loss of habitat, things that they need to actually settle. Sometimes that habitat is just not there or it's decreasing. And then a little more recent, in the past few decades, people have been realizing that warming as well as acidification is really affecting the ability of these shellfish to produce shell. They have to have the right seawater chemistry and temperature conditions to really build that shell on themselves. So these are all things that threaten those important species. And unfortunately, this is sort of a little doom and gloom in the talk. There are expectations some people have that we may collapse our shell fisheries in the next 30 or so years. So one of the tough things about this is that we know what humans take out of the ocean in terms of shellfish, but we don't know what the natural predators actually do. And that's particularly important for the large mobile species that are really hard to track. And so not knowing how much they eat and contribute annually to the mortality of a shellfish, we can't really track a population or estimate its trajectory unless we have that information. And so we do know that lots of things eat shellfish, and this can be problematic, right? So things like otters and rays have unfortunately surfaced in the news lines, kind of like you're seeing here, to where people have wanted to control these animals because they think that their consumption is problematic. I have my own opinions about that, but it just goes to show you that understanding what these animals actually do and how much they eat is really important, because some of these headlines are unfortunately very misinformed. And so who are the animals out there that actually eat shellfish and eat really hard-shelled shellfish? So these are what we call the durophages. That literally means hard eating. So we've got in the marine environment things like rays, other fishes like these sheepshead, sharks, marine mammals like otters, like you saw earlier, but also sea turtles, other invertebrates and crustaceans like these crabs and octopus too. So if we take it a little closer to home in the greater Caribbean, so that includes Florida, the Gulf of Mexico, Bahamas, and throughout the Caribbean proper, these are the main players in shellfish predation. So things like spiny lobster and stone crab, puffer fishes, trigger fishes, various rays, as well as sea turtles. And you're probably thinking, well, how do these animals actually break open a shell, right? When you guys go to a restaurant, it's either open for you or you get to, monkey with it after it's been steamed for a while, right? So it's a lot easier, but these guys are actually pulverizing live organisms that are trying to keep their shell together. So they do this with specialized morphological appendages. And so for crabs, they have these kind of crusher claws. Species of fishes like puffer fishes and rays have these flattened dental plates and really fortified jaws that can really deliver a really powerful blow. And then you got sea turtles like loggerheads with very strong and very robust beak that they can use to even crack open adult queen conch, if you can believe that. But studying these animals, particularly the larger ones, is really challenging, right? They're highly mobile, so that means they're kind of rare, right? You can't just jump in and expect you're going to land on a sea turtle. And because of that, we have really little information about their fine-scale habitat use. In particular, where are they foraging? And also the conservation status of some of these animals doesn't help either. We have very little information on their diet, so most sea turtles are protected, and we really only get information from stranded animals. We're limited in what we can do with live animals. And all of this really comes together to reduce our understanding of what their foraging effects are on shellfish populations. So benefits to working with really large mobile organisms like this is that they're large and conspicuous, right? So you can see them from remote sensors like drones and things, right? Then they also leave feeding traces, right? Because they forage in sediments and are large and sometimes uprooting things out of the sediment, they leave marks like these. So on the bottom left here, this is from Little Jim Flat, just south of Harbor Branch. Because you guys can kind of see that big pothole in the sediment there. That's from an eagle ray foraging on the flat. The eagle ray was foraging, he swam away, and then we ran in and took a photo. So we know it's from an eagle ray. So they'll leave impressions in the sediment like that. And then sometimes you'll get a little bit of messy eating, right? So these guys will leave a little crumb trail of their foraging activities where they'll regurgitate or only partially destroy some of these shells, and you can use that to track where they've been feeding, right? And they also leave traces that you can't see, right? So our group is starting to get involved with environmental DNA right now. We're looking at how animals actually shed DNA during this process. But something you may not have thought about are traces that you can hear. And that's what leads us to our premise today. This idea that this process of breaking open a shell is conceivably audible. At least that's what we thought going into this. And, we were excited about this, and we knew that in the paleontological record, there are folks who've been using this as a proxy for predation. They look for broken shells like this through time in our sediments and try and piece together and reconstruct predator-prey interactions. So why couldn't we do it in real time? And so the idea was, what are the tools out there to capture this? And that's what leads us to passive acoustics. So passive acoustics is a process where we basically put out underwater recorders and listen. And so what you're seeing on the right here, this is a diver installing a recorder. It's got a hydrophone on it. This is basically an underwater microphone. You can just speak into it or blurb your bubbles into it, which we like to do sometimes. And on board is basically a bunch of batteries in this sort of canister, as well as a really precise clock. And so the great thing about acoustics is that it doesn't take a lot of power, right? And it can last, these sensors can last for a very long time. You can do things in day, you can do things at night, right? You don't require any clear water, like say a video or a submerged camera. And then because we have that clock in there, we can relate some of those sounds back to real time. So we know exactly when it happened. And people have been using this technique and technology for a long time now to detect and classify things like whale calls, courtship-associated sounds in fishes. That's something we do here at Harbor Branch, and lots of different applications. And we do this by basically taking an acoustic wave file, right, so an audio file, and then we transfer it, we create an image of it. The image here is something called a spectrogram, and the spectrogram basically looks over time, and then by frequency, and then the intensity of the signal at these various bands, frequency bands. So low bandwidth means it's really constrained. High bandwidth means it's really spanning a bunch of frequencies. And so we had this idea back in 2018, and I was talking with my colleague here, Laurent Cherubin, and then another colleague at Mote Marine Lab, Kim Bassos-Hull, and being like, hey guys, maybe we should just record some of this and see what we get. So Kim had these eagle rays around, and we started to feed them different things. And bam, you're hearing all sorts of fun stuff in here. So a little snap, crackle, pop. This talk is not sponsored by Rice Krispies, but that is sort of what you're hearing, all these sort of interesting acoustic and auditory signals. Yeah, so lots of fun. And, we were like, well, okay, this all kind of sounds the same though, right? So how are we going to be able to distinguish what the animals are eating based on how it sounds? And so the first thing we looked at were sort of the number of those cracks that you're hearing, right? And what came out to us immediately was like, hey, these clams, man, they take a long time to process. The animal's sitting there, munching on the clams, spitting out the shells. Things are kind of shattering in their mouth. Whereas these other snails and things, it doesn't take much to really get through to them, or at least once they're initially pulverized. So this showed us that, hey, maybe there's some way for us to distinguish what the animals are eating based on the number of cracks. But then we wanted to take a look at the actual spectrograms like I showed you earlier. So this is a spectrogram of a ray cracking a hard clam. So you can see it's a pretty broadband signal, right? And the warmer colors here are more intense signal, and the cooler colors are less intense. So this little time period over just the course of, a tenth of a second is when that initial fracture happens. And you can see on the right here, this is the same event, and the amplitude of the signal, the strength of it's really strong initially, and then it kind of cracks them into smaller and smaller pieces. So then this started to let us believe, well, hey, maybe we can also not only tell what an animal's feeding on, but maybe isolate a specific feeding event. And so we were comparing between species here. So the hard clams are up top, the banded tulip, a type of snails, on the bottom. And we see these same kind of broadband signals. And we were like, how are we going to actually pull this apart? Because it looks the same to us. And so everything's got these short broadband signals. And how do we separate them? And so we turned to machine learning. This is a form of artificial intelligence. It basically can learn patterns in the data. So we feed the machine or a computer program labeled data. We tell it what it is, and then it basically trains itself to recognize kind of the very nuanced discrepancies between the things, and then we give it new data, as you can see here, and it can classify it. And we, of course, need this because we're generating so much data that we honestly don't even have enough time to analyze it. And so we need the help of these machines. Of course, we do some quality control and screening for a portion of the data just to make sure everything's all square. But this is a hugely important approach to dealing with the sort of big data generation we're all living in now. And so this took a collaboration with some folks from the Department of Computer Science and Electrical Engineering. If you asked me when I was 6 years old, if growing up to be a marine biologist, whether I'd be working with computer scientists, I'd say hell no. But things have changed, and we really require these tremendous collaborations with these really talented people. And so these individuals helped us basically develop a workflow to process these interesting fracture events. And we just had a paper accepted, should be coming out in the coming months, showing how we can accurately classify what an animal is eating, at least this eagle ray, based on the behavior of those cracks. And so with an accuracy of almost 99%. And so tremendous work went into this, but it just goes to show you that this is possible. And so you're probably wondering, well, Matt, that's cool, but you did all of your work in a tank. How realistic is this work? And so we did go out into the field. We simulated the behavior of these animals feeding on these hard-shelled organisms. And we created these signal loss curves to basically figure out how far away can we accurately detect the signal. So you can see here it goes all the way out to 100 meters, so pretty far. And so this was our initial work. This was our first step into the world of passive acoustics. And it showed us that we can track consumption of mollusks. We can potentially identify prey based on some of the acoustic characteristics. We also showed that field extraction was feasible. But this really developed a whole new set of questions and directions, as most science does. And for example, we just worked on one predator species, the eagle ray. Are other shell-crushing species making the same acoustic sounds? That was a big question. And then we can obviously detect it in a lagoon environment out to quite some distance, but can we do the same on a reef environment or other habitats that these species occupy? So there are a lot of questions here, and we pitched this sort of dual-pronged approach to the National Science Foundation in 2022 to basically allow us to deploy recorders in certain habitats where we expect these types of behaviors to occur, but also put the recorders on the animals themselves in the form of a tag, and we'll get to those a little bit later. And so they funded us, and we've been working on this project since about 2022. And so the idea of this is we're basically developing field recordings of different predator-prey combinations, processing those signals using collaborations with computer scientists and electrical engineers, and then using that information to classify predator-prey interactions, and getting to a point where we can actually track these activities over both space and time in our marine coastal environments. And so we have a bunch of research questions. Today I'm going to just focus on these two. That big one, which is, can we actually distinguish predator feeding on the same thing? So I'm going to present some data on these 4 species feeding on hard clams. And then later on, we'll talk a little bit about where and when shell crushing occurs in marine ecosystems. It's also important to mention that this funding source was through the career program at NSF. And the career program is for early faculty, which I was at the time, early career faculty who were trying to get into a new field. So passive acoustics was really new to me. but also incorporate a heavy education component to this. And so we had a lot of different goals over the years where we've tried to enhance marine science graduate education here at Harbor Branch, and then also engage the public with ocean acoustics. We did some work in Bermuda trying to connect the locals with their environment, but we're not going to have enough time, unfortunately, to talk about that today. So let's go with the first research question. How does predator species influence consumption acoustics? And so we went back into the lab and basically extended our feeding trials we did at Mote Marine Lab to other species. So we fed loggerhead sea turtles, hard clams on the top left there. We fed spot-fin porcupine fish, some hard clams on the top right, and then our stone crabs, some hard clams on the bottom left. And in each scenario, we dipped a hydrophone in the water. We tried to standardize all the measurements, and then we, of course, videotaped all of this to help us validate what was going on. And then we brought the information into a analysis program, acoustic analysis program called Raven. And basically that allows us to plot our spectrograms, isolate different events, and characterize and analyze the behavior of those acoustics. We also use another program called Audacity to help us make some measurements of those events. And so a fracture event, there's a lot going on in a fracture event. So here's the kind of the whole event, and you're seeing all these different pulses, and you see even clusters of pulses as well. So we had to start assigning terminology and really getting organized with how we analyze this data. So a shot is basically a series of pulses in a relatively short amount of time. So we went out identifying shots and then measuring a couple of things in those shots. So the highest, where the highest energy was in the frequency spectrum, and also the peak magnitude of the signal as well. So giving us information about signal strength and where it lies in an acoustic spectrum. And this was part of a summer intern project last year, Maddie DeBest. She performed a lot of these analyses. She pulled information from about 100 different events from these 4 different species. And then here's what it sounds like. All right, so this is our crab. And you guys see there's a lot of space there between the signals, right? All right, and here's our loggerhead. It's kind of like somebody cracking their knuckles a little bit, right? Yeah, so this is the stuff we do and listen to and your tax dollars pay for. Isn't that interesting? And here's our porcupine fish. This one's a little more exciting, right? Yeah, he gets in there, and he's violent. And I say he because his name is Walter. He's actually a fish at the Loggerhead Marine Life Center. If you guys go there, say hello. And then the eagle ray you heard plenty of. So what do we do with this information? Well, we take all those different variables and we try and steer the data apart as best we can to figure out how are we going to isolate and distinguish between these predators. So this is called a principal component analysis. All you need to know is each little symbol on there is a different feeding event, and then they're labeled by the predator species in color and shape there. So the loggerheads are in green down here, and then the stone crabs are in red here. You got your porcupine fish in orange, and then our eagle rays are a little cluster here. And so what you see here is separation really on this axis for the stone crabs, right? And what these little overlays mean is basically that's the variable that's driving the differences, right? So stone crabs have very high numbers of shots, but the event time takes really long. The shot duration for loggerhead sea turtles is relatively high. As you kind of heard, I was kind of insinuating it was taking a long time there when we were listening to those acoustic files. And then our porcupine fish has this sort of shot rate, a lot of different pulses in a short period of time. And so with this information, we could really pull apart, who's doing what and potentially who's feeding in a certain area if we're just listening. And so this just kind of recaps what we were able to find to try and distinguish between those 4 species. And so, okay, again, you're probably like, well, that's cool, but it's in the lab. So show us some field evidence that you can actually record this stuff. And so we went out and deployed these things, are affectionately called one-eyed unicorns. And so it has nothing to do with unicorns, obviously, but we have these hydrophones that we were setting up with paired camera systems. And the idea here was, okay, we're going to basically watch and point our hydrophone right at what's happening in front of us. And we did that over in Fort Pierce Inlet. This is, this camera setup. So they're weighed down with these cinder blocks. And in front of them, we put these trays of clams. It was beautifully set up. We set them up exactly 1 meter away from a hydrophone. In the acoustics world, the sound pressure level at 1 meter is a pretty standard measurement. So we wanted to see if we can get that. And we basically tried to bait in the predators to see who would come by and crush things on camera and on audio. And we didn't get anything. Went through all this work, which was very frustrating at first, but that's science. And we decided, okay, let's switch this up a little bit. Let's take the clams out of the trays and let's move location. So we went up to Sebastian Inlet and we dumped the clams right on the sand. And boy, we got a lot going on there. And so you can see some of it here. So just some, fractures and remnants of shell fragments. You can also see some other smaller predators that got to come into the mix, like these 9-armed sea stars, as well as various types of crabs that came in. Okay, and we got some cool audio, right? So this is, we're getting towards the middle of the talk, and to see that you guys are awake, we're going to play a little bit of a game, all right? It's called Who Done It? And I'm going to give you 2 different sequences. One's a crab, and one's a ray, but I'm not going to tell you which one. So let's go ahead and listen. Here comes the first one on the left. Now, mind you, we've got a lot more clams in the mix than we typically do when we're doing these experiments. So we got about 50 clams there. Probably lost about 30 right there. And this one. Okay, so who thinks the ray is on the left? Who thinks the ray is on the right? Okay. You guys ready? Boom. So these are images that we grabbed right at the same time these events were happening, right? So you can see that some of the stuff that we're learning we can really apply here. And this is what we call validation of our technique. So very exciting. And then we took that detection classification method we used in the captive work, and we tried to apply it to this field data. Because all we had out there were clams, and we said, okay, detector, let's see what you got. And it was wrong 60% of the time, unfortunately. But that was just by analyzing a single individual crack, right? And then we told it, okay, well, look, if the cracks are lasting for this amount of time based on our captive experiments, really incorporate into your analysis that sequence, okay? Not just a single crack. And then we got better. We recognized 100% of those and correctly classified them as hard clams. So this is part of that paper, just to show you that we can really do some great things with this. Okay, so where and when does this occur in marine ecosystems? So this is kind of the first part was fun too, but I think you guys are gonna like this part a little more. It's a few more videos. So we can't go out there and put hydrophones everywhere, right? As much as we'd like to, this would be a nightmare. And so how do we, capture where shell crushing predation's occurring? And we're gonna turn now to animal-borne tags, okay? So these predators move around quite a bit, right? However, they're big enough where we can actually put some sophisticated tags onto them. So this is called a CATS cam. It's made by a company called Customized Animal Tracking Solutions. It has nothing to do with pussycats, so that's not what we're talking about. In these tags, which are multi-sensor biologgers, you have depth sensors, temperature sensors, a gyroscope that's kind of measuring the twisting of the tag, a magnetometer that's giving you the direction that the tag is facing, and a triaxial accelerometer. So it's sort of the full limited suite here, high trim level tag. And on top of that, we got a camera. We have a hydrophone, our little audio workhorse, and then we have a recovery transmitter. So we have the recovery transmitter because we actually want this to dislodge from the animal, and so that we can recover the data. And so I'll show you how that happens in a minute here. Just want to recognize Ben Metzger, a mechanical engineer here at Harbor Branch. He's in the crowd somewhere, and he really helped us with this whole tag design. And so the underside of the tag has these suction cups, and we were looking for a really non-invasive way to attach this device to these animals. And the rays we were studying, these spotted eagle rays, have very soft skin. It's almost like a mammal, really, really soft. And so we use these silicone suction cups that basically suck to their head. And then we added on a little bit of an addition here called a galvanic time release cradle. And so what happens is we use these little printed hooks that fit perfectly on the spiracle of the animal. It's actually like an accessory breathing organ, but the top ridge of it is highly cartilaginous and nice and rigid. So we attach that to it with some elastic bands. The elastic bands attached to this galvanic time release. This thing corrodes over time. This one's a 24-hour version. Sorry, so excited I'm dropping the mic here. And so that'll corrode and basically release the straps. And the suction cups only hold for about a few hours, and after which the whole thing slides off the animal, and then we can recover it. And so this was just a quick clip from Changing Seas last year. I don't know if anybody caught this episode, but they filmed us doing this work both in Bermuda and Florida. It was really fun, and I'm going to show you how quick the attachment process is. So wipe, suction cup, put on the band, and that's it. She's ready to go. And I can say she because this is a female ray. And so over the years, we've been attaching these to animals between Bermuda and Florida. Like I said, we really started our work in Bermuda. We figured, we'd start with clear waters and so on. As it turns out, things were much more productive locally here. And did some work here in Fort Pierce as well as over in Sarasota. And this is sort of the size range of the animals. So smaller animal down to about 1 meter wide, and then this beast, which could almost take me. And so we get these tags to last typically around 24 hours. Throughout all of the deployments, we've accumulated almost about 490 hours of recorded data on these animals. So pretty extensive data set. And these are the locations where we deployed them. So Sarasota Bay, a little bit off, and this inlet here, New Pass, a little bit offshore. Fort Pierce around the Tucker Cove area, also kind of down here off the Smithsonian. And then in Bermuda, in Harrington Sound primarily, as well as Flats Inlet. And really the brainchild behind all this is Cecilia Hampton. You're going to see her quite a bit now and towards the end of the talk. And so we also overlaid the available bathymetry. So this is basically the depths of the areas, all right? So warmer colors, these are shallower waters. Deeper colors, these are cooler colors, these are your deeper waters. So you can see off of Bermuda, you got a lot of deep water pretty close by, even in these inshore sounds and lagoons, whereas here in Sarasota Bay, as well as the IRL, a lot shallower habitat. So what's cool is that really no matter where we've worked, Bermuda's in red, I think Sarasota's in blue, and the IRL's in green. These animals have shown very consistent behavioral patterns. So this is basically depth on the y-axis. Surface is up here, bottom down here. And this is time of day. So noon would be here, and nighttime would be there and there. So really during the day, they're diving deeper, is what this is basically showing you. But it's very consistent across the different study sites. And here's what the footage looks like. So you get this kind of ray point of view. And we've had this incredible opportunity to work with a NSF Learn student, Ethan Canto here, who's from the Wilkes Honors College. And Ethan really helped us with the video labeling. So we go straight to the video first because we know that has the strongest validation potential for us. And so Ethan will go through and make sure there's, so in this case, the bottom is visible. He'll annotate when the animals are swimming. So in the top two, you can kind of see the animals and the ground or the seabed moving underneath them as they're swimming forward. They'll also point out when there's another ray in the picture. We call that a conspecific, which is another individual of the same species. You can see the tail of the ray there. Hopefully you guys can see that. And then most pertinent to this project, whether the animals were foraging or not, which you see down there on the bottom right. And so Ethan goes through all these labels, and then C kind of takes all that information and puts together incredible videos that we see here. So here's the animal kind of swimming with its tag on, and here is the ray's point of view. So what we're showing you here is the animal kind of swimming around. On the top, this is the depth, right? So the animal's right now kind of testing the seabed. Maybe it's smelling things, maybe it's feeling things, like electrical activity, for example, in the sediment. So sharks and rays have very specialized organs where they can really detect weak changes in electric potential. So we suspect they might be doing that. And then the depth track is kind of tracking those sort of up and down movements that you can hopefully see there. And this is also cool because it gives us a lot of information about the habitat. And look, the ray's found something now, and now it's starting to do that real digging behavior, and it's creating a giant smoke screen, right? And this is resuspending the sediments, right? And this is actually a very important ecological process. People have at least hypothesized over the years that this is what helps bring and aerate sediments. And now the ray picked up something, and he's starting to crunch on it. And you can see during this process, the ray is actually swimming up while he's doing this. We don't know why they do this. We see little variations in this behavior, but from time to time, we'll see them come off the bottom, and maybe they're a little vulnerable when they're at the bottom, so they want to kind of, while they're chewing and crunching their clams, make sure there's not a shark following them or something like that. And so, yeah, and then this animal will finish crunching this thing up. You can see it starting to bank down, probably back to where it found that prey, and maybe go back to feeding. Okay, and so this is that same event, a time series of it. It was only about 2 minutes, in case anybody was counting. And so we can go ahead and overlay the labels of what the animals are doing with their depth profiles, right? So here's, again, the animal coming up out of the sediment, where it starts swimming with the whatever prey was in its mouth. And then we can also plot things like the overall dynamic body acceleration, or ODBA. That's a real integrated measure of their, basically how much acceleration is happening. And then we can break it down into acceleration in 3 axes. So the stars here are basically when the animal was processing that prey, right? So you could see we're actually picking that up on the accelerometer too. But we've got that hydrophone as backup, right? So we can pull apart some of these events and acoustically assist our analysis. And so pretty amazing. This is the first time anybody had done this, and Cecilia was able to publish this paper last year, showing basically how we can integrate these various sensors to study the foraging ecology of these animals. So that was based on videos in Bermuda. I'll take you a little closer to home. So this is Fort Pierce Inlet last year. A little murkier, as you can see. But here's a large ray who's digging in the sediment, and she's not going to really find much here. And so she starts to kind of move away. But you can kind of see she kind of senses something, and you're about to see the scaredest crab in the world there. And that was a hermit crab, and the hermit crab is now in the ray's mouth. The ray actually manipulates this for some time. Maybe the crab's fighting back in the mouth. We don't really know what happens there. But eventually, the ray wins. And if you thought that was sad, let me show you something a little sadder. So this one was interesting, right? So this is over in Sarasota. This is another large female ray. And yeah, you'll see a couple of things here. And there are some conch that are copulating, doing their thing, and yeah, she changes that. So anyway, we get very excited when we hear things getting crunched in this lab, but it's not exciting for everybody, clearly. All right. So, and that's only when we have the camera on, right? So when we put out these tags, we don't have the camera running through the night because we can't see anything. And so we actually turn it off, right? But we can still rely on, our knowledge of acoustics, to recognize when some of these feeding events are happening. And actually, it's very subtle here, but you can kind of see some of these kind of rhythmic, low frequency bumps here. That's the ray actually excavating the sediment, we've learned over time. And here's the ray starting to get into something. Yeah. All right, so where and when is this happening? So here are all of our events that we've compiled so far. So we've recorded over 250 confirmed feeding events. And what you're seeing here is, again, a scale of depth from shallow surface down to about 8 meters, and then time of day. So daylight is here, nighttime is here and here. So you can kind of see a whole cluster of things going on in the early evening, which is really exciting. And yeah, so the rays are feeding on infauna and epifauna, mollusks and crustaceans. Everything we've seen them feed on the camera have been things that we've actually extracted from their diet in other studies. So there's nothing artificial happening here, it seems. And then we see these interactions happening from really shallow waters. We suspect almost intertidal waters where maybe sometimes the tag actually seems like it's getting into air. So the animals might be foraging in just a few inches of water, all the way up into through the evening time. So we're learning a lot with these animal-borne sensors. And as far as where these interactions are occurring, we're a little crude on that right now. So this is called a pseudo track. So that's Fort Pierce Inlet there. Harbor Branch is right here. You guys are sitting right where this pointer is. And so it's a little inaccurate right now. This is a pseudo track that's based on dead reckoning, right? So we know how relatively how fast the animal can swim. We know what direction it's heading, and from that we can kind of piece together maybe where it's gone. And this summer we're going to have a telemetry array up that's going to basically help us localize where the tag is and basically pin and staple these tracks at known locations and times. So we expect we'll have a lot more accurate data moving forward. And so if you guys see one of these out on the lagoon, we want it, dead or alive. It's very important. It's got a lot of information on it, and it's very expensive. Okay, so research-wise, I think we've seen some really cool things. On top of this, don't have time to talk about it, we see a lot of other interesting behaviors we don't think anyone's seen before, both intra-specific, meaning eagle ray to eagle ray, is also between eagle rays and other organisms, not just their prey. We document some human interactions, so we know when boats are by based on the audio. We saw one ray almost get run over by a boat on the camera, which is a little scary. And then, seeing them move between lagoon systems and all the way out to the offshore reefs demonstrates, I think, the connectivity that these animals are capable of as well. But we're here for the foraging and the predation on shellfish, so let's focus on that. And we're developing the first ever foraging rates for these species, really learning to better understand what their impacts are on these populations. And that's not only important for understanding what habitat the predators require for their own conservation and management, but also how much mortality they're inflicting on shellfish populations. So this summer, we're going to do some more enhancement experiments, again, where we plant bivalves in front of a one-eyed unicorn, and we're going to use that to help us develop and train more of our detection classification schemes, so we can now strip out, based on long-term field data, what's going on and who's being eaten by whom. We'll also be doing some more animal-borne tagging this summer. So again, if you see the tag, call the number on it. And with rays, and then we're expanding to loggerhead turtles as well. Another formidable shell-crushing predator. So this Dr. Liberty Boyd there is going to be running all that. We also got some new funding. So the state of Florida has this major initiative, in case you haven't heard of it, to restore clams, including the Indian River Lagoon. Probably heard of that. And they're doing this experiment in and out of seagrass beds. So they're going to dump clams in the seagrass beds. They're going to dump clams in the sand nearby, and we're hopefully going to record everything that gets eaten. Okay, so as I said, we have some major educational goals here, and one was enhancing marine science and education at FAU Harbor Branch. So you've seen Cecilia show up in these various videos. So she's the Ph.D. student we've been training on this project, and she's been getting a really awesome cross-disciplinary training. She's taking courses in machine learning, various marine science and oceanography courses. And we couldn't have done that without our great collaborator from Woods Hole, Dr. Aaron Mooney. These are some images of our trip where he hosted us for a workshop a few Octobers ago. You can kind of see us, deliberating how to deal with our data, learning about new tags. These are smaller biologging tags that you can attach to squid and other things, as well as how his team is processing data. So a lot of great opportunities for graduate education there. And as part of this course, we've also developed a marine trophic ecology class at FAU to help expand our curriculum. So these are some images from 2024. We really try to emphasize experiential learning and practical learning. And so these individuals really got some great experience there. This is our 2026 class, which is actually over here. So wave, guys, yeah. So these are real students. This isn't just a model photo thing. Yeah, so really great opportunities to work with students and train them in this really kind of broad field. But last and certainly not least is engaging you guys with ocean acoustics. So hopefully you guys learned something today. And if you haven't or need more or just so enthralled by it, check out the Discovery boat tour. So as Gabby said, this thing's hooked up outside. We integrated a hydrophone this year into the programming and worked with this great intern over the summer, Isabella Holden. And Bella did a tremendous job learning the nuances of this kind of sampling technique, and they were really able to integrate it into various programs, visiting school groups like you see here. They have these awesome screens where you can kind of learn about the different animals and the different sounds that they're making. So it's not just all about shell crushing predation. There's a whole bunch of other sounds out there in the sea. So you can learn a little bit more about that there. And they even took some time to study how the students were affected by these, how their learning was affected by some of these experiences, and asked them, do you know what a hydrophone is? And at the onset of the field trip and then also afterwards. And so the students are learning. They do know what this is, and that's great and encouraging. We also trialed this with 48 guests over the summer and got some feedback from them, and they really enjoyed it. Since then, we've actually, Gabby just told me this afternoon, we've had over 1,000 people engaged now by this hydrophone integration, and everyone's really enjoying this. So definitely check it out. These are some fun quotes that people had. People like the pork fish because they snort like pigs, and I think people are genuinely interested in hearing what's going on underwater. And I recognize being able to experience that world is not accessible to everyone, and this is a great way to do that. And Gabby's team really made that possible here. And so we're working together now on an exhibit at the Visitor Center. So hopefully when you guys come here next year, audio waves will be up, and this is an interactive exhibit. You can see we got some headphones in there and a digital tech panel that you can kind of blast through. And these are some of the education staff who were testing it out last summer. So we have to make some modifications to it, but we also got some, cool little models of shell crushing jaws and the shells themselves and all sorts of fun stuff. Okay, so this was a tremendous effort, all of this work, and I definitely didn't do it alone. I tried to highlight people and collaborators, but that was, just a subsample of everyone who made this possible. So I really want to thank the funders in particular, so National Science Foundation for taking a chance on me, and the Harbor Branch Oceanographic Institute Foundation, who provided seed funding basically to pilot this work and really made this all possible, and I think allowed us to get the NSF money, excuse me. And so we also have great staff and facilities here. I mentioned our engineers, but also aquaculture staff who can help us, do all sorts of fun stuff in aquariums. And our great partners in this project, particularly Moat Marine Laboratory, my colleague Kim, and my colleague from Woods Hole, Dr. Aaron Mooney, and other groups who helped us get access to animals and facilities, such as In-Water Research Group down in Jensen Beach, and then in Bermuda, the Bermuda Aquarium Museum and Zoo. And we're just starting to work with the Shed Aquarium on some queen conch mortality stuff. So I didn't have time to go over that today, and it's kind of still developing. So we're hoping to present on that sometime in the future. And last but not least, my wonderful lab of students who've supported this work, whether it was their project or not. Everyone kind of got involved, and really appreciate that. And then certainly not last but not least, my wife Rachel, who has dealt with me and my crazy ideas and spending time working on these proposals, and just been a really good supporter as I've moved through this process. So, yeah. As I say, behind every man. Yeah. Okay, and so check us out on Instagram. That's our handle there, Fish Eco HPOI, and I think we'll take any questions. Thank you.
All right, great job, Matt. Great presentation. Always good visuals and sound files too today. So great job. Okay, awesome. So we'll take some questions. We have time for a couple. I'm going to repeat them for those listening online, or at least paraphrase them. So let's start right in the front row here.
How about the correlation between predator and prey? What I'm thinking about is when shell life declines. Long Island, eastern Long Island, used to have scallops, lots of scallops. And you mentioned the blowfish, the porcupine fish, the great predator for shellfish. Right around the same time scallops decline, blowfish decline. They were probably eaten, what you're talking about, right? So you might have to answer that.
Great. So for those online, the question was about predator and prey interactions and how decline of the prey could affect the population of the predator.
Yeah, so that's a great question. I'm from Long Island originally, actually, so somewhat familiar with the bay scallop declines there. There are other sources, right? So some folks think water quality and habitat loss, particularly seagrass, has also played a role. But certainly, it kind of highlights that we also have to be watching these shellfish prey for the predators themselves too, because they are reliant on them. Now, as far as how they, may have the puffer fishes may have affected the scallops, I don't quite know, but it's certainly a possibility.
Great question. All right, next up. All right, good question. Can your research be used to predict shellfish populations and maybe the fishing capacity or carrying capacity for human activities?
Yeah, that's where we're hoping. We're hoping to at least get a little more information on, what these animals require, in terms of a daily basis. A lot of these animals, when you keep them in captivity, we found, at least with the eagle rays, we can't keep up once they start feeding. Like, it's hard for us to actually give them enough food. And they start to lose a little bit of weight, right? So, hopefully some of these like animal-borne approaches will get us to that. And it is an important aspect. We're not the only people or the only organism on Earth reliant on these shellfish populations. So we need to be thinking about, again, the animals that are reliant on them too. So I'm hoping we'll provide some of that information. Maybe get some more funding.
Let's see. Did you have a question in the jacket?
How long ago were those shells alive? I mean, did something
When you find a shell on the beach, how long ago was it a home to a living animal?
Yeah, so it depends. You know, you know, a colleague of mine who studies, who's a a malacologist, basically studies mollusks. He says that a lot of those shells can retain their color and luster. I mean, sometimes for decades. So it's really hard to predict how long ago that was. There's other information and analyses you can do, like chemically, to maybe understand how recently that thing died. And that's something we're hoping to actually get into with some other research. But yeah, it can be a wide range. It could have just happened, and it could be, 20 years old. But a lot of it depends on, the mollusks have this thing called a periostracum, and that's sort of like it's like almost like a sheath that's on the shell itself. And if that's there, then you have a pretty good indication it was pretty recent, because that doesn't last very long.
The other thing I wanted to ask is stone crabs. My understanding, and I may be wrong about this, is that now they can break off both claws for a stone crab, and used to be they would get one and it would grow back, right?
All right, the follow-up question is about stone crab claw harvest, and traditionally it was harvest one claw, leave one for the crab. Now what happens if they take both claws?
Yeah, so good question. So yeah, there were a lot of studies done, once the regulation came out about obviously removing one claw, and the animals were able to survive, but maybe not as well. As far as removing both claws, I imagine that they can still survive, but probably it's not a good situation. There probably limits what they can feed on in that process.
Probably good to leave them a claw wherever possible. Okay, next up.
Now that you've been doing the studying, have you found that like the people raising clams and mollusks and everything are starting to say, well, how can we stop them from eating them?
How have your studies helped people growing different shellfish prevent predation?
Right. So one of the things we'd like to do and offer as a product from this research is kind of a detector or alert system. When animals are in a certain area, we can hear them like crunching, so to speak. Basically, kick off a series of events, that basically deters them. Right. So we have a lot of ideas on that. But most of our research to date has been on working on keeping animals out of the system to start with. Right. So that's sort of where we're at with that. We've been using various types of deterrents with some colleagues on main campus, magnetic deterrents that we're testing, as well as electrogalvanic deterrents. And so it certainly has relevance to the shellfish industry and those involved with aquaculture and restoration. So we're definitely making those connections and seeing how we can support those activities.
Great. All right. Well, okay, our final question in the back. Yes.
CAT scans,
Yeah, what's the cost of one of your tracking devices?
they're expensive. Yeah, 10 grand.
Wow.
10,000 bucks. So the guts of the device is about 8 grand or so. But then when we put on all the other accessory things, the float, we pay for folks to help us craft them and all that. Winds up being closer to about 10,000 bucks. So that's typically what we budget. Now that doesn't include either what it takes to go out and deploy those things, right? So we need people to do that, catch the animals. We have to pay for satellite time. So when the transmitter comes up and starts talking to us, we gotta pay for that. So it's very expensive, and yes, we're always looking for donations, if you're wondering.
Awesome. Well, before we thank our speaker, after this, Matt will be in the gallery, and we'll have some cookies. You can ask any additional questions or decide if you want to fund a pop-up tag for us. But thank you all so much for being part of the series. I'm going to miss seeing you all every week, but we hope that you'll come and check us out at the Visitor Center, participate in our tours, come to the Film Fest. It's going to be awesome. We still have to plan it, but it's going to be so good. And we'll see you all back next season. Make sure you fill out that email survey and give us your feedback. And with that, thank our speaker and all the staff who helped.